Okay, today we're going to talk about metabolism and energy. So, looking first at metabolism, think about what comes to mind when you think about metabolism. When you hear the word metabolism, what do you typically think about? Most people think about energy or food, but the definition of metabolism is all of the chemical reactions that are taking place in an organism. So it is all of the chemical reactions happening right now in your body. So it is kind of the emergent property of life that arises from all of the interactions between molecules within the cells. And obviously, we need all of these reactions to take place in our bodies. So when we think about metabolism, we kind of need to understand that there are metabolic pathways that are happening in our body. So a metabolic pathway is going to be a step-by-step process that happens. And so it's going to be one specific molecule that will go through a series of steps to get to the product. So most reactions in the body are metabolic pathways. There's a series of reactions that have to happen to convert one molecule into another. And typically, each of these steps is catalyzed by a specific enzyme. So if you've got starting molecule A is converted to B by enzyme number 1 in the first reaction. B is converted to C by enzyme number 2 in a second reaction. And then C is converted to D in a third reaction by enzyme number 3. So there are metabolic pathways that are step-by-step series of reactions that happen with enzymes that are catalyzing each of those steps. And there are two types of pathways, two types of metabolic pathways. There is a catabolic pathway, and with a catabolic pathway, you are releasing energy, and it releases energy as you break down complex molecules into simpler ones. And cellular respiration would be an example of a catabolic pathway where you're breaking things down, and in that process, it releases energy. And so cellular respiration breaks down glucose, and as the glucose is broken down, energy is generated. And one way to think about or remember catabolic pathways, think about cats. They're very destructive and they release a whole bunch of energy as they tear and shred things apart. So they'll break down your couch and they'll shred your bags. They're very good at destroying and breaking things down. So think of a catabolic pathway, releasing energy and breaking down molecules. The other type of pathway is an anabolic pathway. And anabolic pathways use or consume energy to build complex molecules from simpler ones. And so when you're doing protein synthesis, you are using energy to put amino acids together to build those proteins. So that would be an example of an anabolic pathway. And one way to remember an anabolic pathway is think of Anna from Frozen. She's building her snowman and takes a lot of energy to put together that snowman. So think of Anna building a snowman as an anabolic pathway. Okay, and then there are different types of energy, different forms of energy, and we're not going to get into that too much. That's more of a physics topic. But there are different forms of energy, and it is kind of important to know some of the basics when you're talking about energy. So energy is simply the capacity to cause change. So energy is going to exist in different forms, and they're going to be performing work. At least some of them can perform work. So kinetic energy is going to be energy that's associated with motion. So kinetic energy is the energy of motion. Potential energy is stored energy because it has potential to do work. And so because of its structure or its location, it has potential. And then chemical energy is going to be a type of potential energy that is available in chemical bonds and chemical reactions. So chemical energy is going to be potential energy available for release in a chemical reaction. And so that's kind of the chemical energy is really important for living things. And so that's kind of what we will see as we move through this unit. And energy can be converted from one form to another, but it cannot be destroyed. And so that's kind of where the laws of thermodynamics comes from. And there's two laws of thermodynamics that we kind of need to touch on. We're not going to get into that too much. Again, that's physics, but thermodynamics is simply the study of energy transformations. And the first law of thermodynamics says that energy can be transferred and transformed, but cannot be created or destroyed. So that's the law of conservation of energy. And we're gonna see that in cells and within the body, but we'll also see it later when we talk about the ecosystem and energy transferred from one trophic level to another, but we'll come back to that a little bit later. But energy can be transferred, so it can be converted from solar energy to chemical energy to heat energy. So we'll see all of those transformations of that energy. And according to the second law of thermodynamics, every energy transfer or transformation increases the entropy or disorder of the universe. And we'll kind of touch on that a little bit, but we're not going to get into that too much. But this picture kind of cartoon shows exactly what entropy is. Another thing you can also think of with your room at home is it might be clean on Sunday, and then as you go through the week, it gets dirtier and dirtier, messier and messier. You don't have time to hang up your clothes. You don't have time to make your bed. Your laundry is piling up. Your room goes from a very ordered state to a very messy, disordered state. And it takes a lot of energy to clean up your room and put it back into order. And so that's kind of what, when we think of entropy, we think of order in the universe, and things tend to naturally go from an ordered state to a disordered state. And so it takes a lot of energy to maintain that order within the universe. And biological things also need to put in some energy to keep those things in order. And so it takes a lot of energy to become and maintain order within an organism. We also need to look at free energy, and there is a formula for free energy, but we're not going to get too much into that formula. But what we do need to know is what the negative delta G or negative free energy means, and what a positive delta G or positive free energy means. So delta G is simply your change in energy or change in free energy. And when we have a reaction that is going to give off energy or a reaction that's going to require energy, that's where we're going to see the positive and negative delta G. So an exergonic reaction is going to be a reaction that proceeds with a net release of energy. So it gives off energy. An exergonic reaction gives off energy. And an endergonic reaction is going to be one that absorbs energy from its surroundings. So it's going to use energy or need energy. And so if we look at an exergonic reaction is going to have a negative delta G. And so it's going to give off energy. And so an exergonic will be negative delta G. And then an endergonic reaction is going to be one that requires energy to go in, and that's going to have a positive delta G. And ATP is going to be one of those that will give off energy. And so the breakdown of ATP is an exergonic reaction. And we can actually couple that reaction, that energy that's given off from ATP, with other chemical reactions that require ATP. And that coupling of chemical reactions is kind of how our cells work and function. So ATP is the molecule that cells use, the cellular molecule for energy at a cellular level. And we can again couple chemical reactions, the breakdown of ATP, to drive other reactions in the cells. If you remember hydrolysis back from our first unit, when we're talking about breaking down and putting together macromolecules. Hydrolysis is when we are breaking apart molecules. So in the biochemistry unit, we talked about taking apart macromolecules, and that's hydrolysis, and we have to put the water back in. Well, when we talk about ATP, we are doing hydrolysis of that ATP molecule. And the energy, again, at the cellular level is ATP. That stands for adenosine triphosphate. And that energy is stored in the phosphate groups. So if we look at it, the structure of an ATP molecule, it looks very much like a nucleotide, but it has 3 phosphates instead of just 1 on it. And so the energy is in the phosphate groups. So when we do hydrolysis and we remove one of those phosphate groups, energy is released. And that energy can be coupled with other chemical reactions. So ATP will hydrolyze and form ADP. And of course, water has to go in to replace those bonds to make those molecules stable, and an inorganic phosphate group. So ATP is converted into ADP and inorganic phosphate, and typically you'll see that with a P sub I, and then energy is given off. And so in that hydrolysis process, energy is released. And the energy is released from the ATP when that terminal phosphate bond is broken. And this energy comes from the chemical change to a state of lower free energy, not necessarily the actual breaking of that phosphate bond itself. But just know that when that phosphate group is hydrolyzed, energy is released, and we can couple that energy. And typically what will happen is that inorganic phosphate molecule, think of that kind of as a bundle of energy. That's going to get transferred to another molecule, and that transfer molecule, the one that picks up that phosphate group, is called a phosphorylated intermediate. It doesn't keep that phosphate group attached onto it, but while it has it, it kind of is a bundle of energy that will energize that molecule. And so if we take a look at how we can couple that, in this first reaction up here, glutamic acid plus ammonia can be converted into glutamine. But that's going to require energy. So it's going to have a positive delta G, and it's going to require 3.4 kilocalories per mole of energy. The breakdown of ATP, as the ATP is hydrolyzed, it actually will release 7.3 kilocalories per mole of energy. So it has a negative delta G of 7.3 kilocalories per mole. And because it's giving off more energy than this reaction requires, we can couple them together. So in this second picture here, you're seeing glutamic acid and ATP. If you hydrolyze that ATP and transfer that phosphate group to the glutamic acid, that becomes a phosphorylated intermediate. Then the ammonia can combine with it because it's now been energized, and it will form the glutamine. And as a result, you'll also have ADP and inorganic phosphate left over. This we can recycle, and we'll talk about how we recycle that later. But the glutamine is now, that reaction is using the ATP energy to form that glutamine from glutamic acid and ammonia. So the glutamine plus the ammonia, that requires 3.4 kilocalories per mole, and the hydrolysis of ATP is giving off 7.3 kilocalories per mole. So in the end, we have a net of negative 3.9 kilocalories per mole. So we're actually, there's more energy given off from the ATP than is required for that reaction, so we can actually couple it. And there's enough energy here to drive that chemical reaction. So that kind of is an example of how we're coupling the chemical reaction, the breakdown of ATP, to drive other chemical reactions in the body. As long as the energy given off from the ATP is greater than the requirement of that reaction, you can couple those together. And so there's lots of reactions in the cell that we are doing this with. So we've seen it in our transport proteins. We can see it in our muscle contractions, transport, and muscles, mechanical work. All kinds of reactions require ATP and are using ATP in a coupling scenario. That ATP molecule can then be regenerated. We can actually put that inorganic phosphate group back onto the ADP to make ATP. And that is what cellular respiration is all about, the regeneration of the ATP. So it's a cyclical process. So ATP plus water can be hydrolyzed, and that can be giving off energy necessary for cellular work. The ADP and the inorganic phosphate can be put back together, and that process will happen in cellular respiration. And we'll take a look at that in another video. So ATP, the breakdown of ATP is giving off energy necessary for cellular work, and then cellular respiration can put the phosphate group back on the ADP and recycle the ATP. So we say it's a cyclical process because we're recycling the ATP molecule. Okay, so that is your metabolism and ATP structure. And then we're also going to take a quick look at enzymes. So if we think about the type of macromolecule an enzyme is a protein. And it is a special type of protein. It is a catalyst. And a catalyst is a chemical agent that will speed up a reaction without being consumed by the reaction. And so an enzyme is a protein catalyst that will speed up chemical reactions. And it typically, when you're looking at vocabulary and endings, an enzyme is typically going to end in the ending ASE. And that ASE typically comes behind the prefix that goes along with its substrate. So if we look, for example, here at this picture at the bottom, sucrase, ASE, is going to be one that breaks down sucrose. So sucrose is its substrate, and its products are glucose and fructose. So in naming enzymes, the prefix of it often comes from its substrate, and then enzymes always end in ASE. And how they work is they lower the activation energy necessary to get a chemical reaction to go. And typically you'll see it as E sub A for activation energy. And so typically when you've got chemical reactions, whether they are exergonic or endergonic, if they've got a delta G that's positive or negative, they're all going to require some kind of energy to get them going. So even if you've got a chemical reaction that's giving off energy, it still needs a little boost, a little push to get that molecule, that reaction going. And that energy is called the energy of activation, or the activation energy. And so in this picture, here is the amount of energy necessary to get that reaction going. And what enzymes do is they reduce that activation energy. They lower that activation energy barrier so you don't need as much energy to get it going. And so this is how much energy, much less now, that you need once you've got an enzyme that helps that chemical reaction to go. And the analogy I like to use is, imagine that there's a big boulder sitting at the top of a hill. It's not going anywhere, but you know it could simply roll down that hill. But you're going to have to push it to get it to actually roll down that hill. So imagine you're at the top of the hill and you're pushing this boulder to get it to roll down the hill. How much energy you have to use to push it to get it to roll down the hill, that's your activation energy. But now imagine that you're at the top of the hill with your best friend, and now you're both pushing it as hard as you can. You don't have to push it as hard if you're doing it with a friend than if you're doing it by yourself. So how much energy you have to push it when somebody's helping you is much less. And so that is what an enzyme does. Your best friend is kind of like the enzyme. You don't have to push it as hard if they're also pushing it with you. So it reduces the activation energy necessary to get that boulder rolling down the hill or get that reaction going. If we look at how enzymes work, there is a substrate that's going to be the molecule that it will work on. And once that substrate fits into the active site, which is the location on the enzyme where the substrate fits into, that enzyme and substrate will form an enzyme substrate complex. And induced fit of a substrate brings the chemical group of the active site into a position that enhances their ability to catalyze the reaction. So basically, when that substrate is in the active site, induced fit allows it to arrange the molecules so it reacts best, and it speeds up the chemical reaction while it's in that enzyme substrate complex. So if we look at this picture, the blue is your enzyme and the red is your substrate. So your substrate is going to fit into this active site. It's kind of like a lock and key. It's very specific, and the shape of the enzyme determines its function. And so the substrate is going to fit into that very specific shape. And once it's in there, it will form that enzyme substrate complex. And how an enzyme works in catalyzing reactions is it's going to cause several things. Several things could happen that will actually allow it to lower the activation energy. It could lower the activation energy by orienting the substrates correctly, or it could put strains on the substrate's bonds that allows it to react. Or it could just simply provide a favorable microenvironment for that reaction to take place. Or it could covalently bond to the substrate, allowing the reaction to take place. So different enzymes are going to work differently. It's going to be one of those things that's happening as that substrate is in the active site. And here's just another diagram showing you the substrates are in the red and the green. They will fit into the active site, and then while they're in there, you've got your enzyme substrate complex. They will react inside the enzyme, and then they will release the products. The enzyme is recyclable or reusable. Some enzymes will put things together. Some enzymes will take things apart. Some enzymes will just convert one molecule to another molecule. Each enzyme has a specific job to do, and its structure will determine how it functions. And so the substrate will fit into a unique active site of an enzyme. Okay, and there are conditions that will affect the activity of the enzyme. So it will interfere with the activity of the enzyme. So the environment that it's in will have an effect. The temperature and the pH are typically the most common factors that will affect an enzyme's reactivity. And so those chemicals in the environment will definitely have an influence on the enzyme's reactive site and reactivity. So each enzyme is going to have what we call an optimal temperature, which it can function. So it's going to have the greatest reaction rate. It's going to work the best at its optimal temperature. It will also have an optimal pH. Again, the pH that it works the best at. So the optimal conditions are going to favor the most active shape for the enzyme's molecule. So again, structure equals function. And so the environment is going to influence the structure of the enzyme. And so the optimal temperature and pH, the optimal environment, is going to make it work the best and have the highest reactivity. And so if we look at temperature first, each enzyme is going to have an optimal temperature. Some enzymes work great at lower temperatures. Some enzymes work at higher temperatures. If we look at body temperature, human body temperature is 37 degrees Celsius. So there are the enzymes that we find in our body would make sense that they are going to work the best at 37 degrees Celsius. And so human enzymes work best at 37 degrees. If we look at other enzymes, let's say a thermophilic bacteria that lives in the hot springs, they're going to have enzymes that work best at 77 degrees Celsius, very high temperatures, because that's the environment that they thrive in. And if we look at what will happen if we change the temperature, if we look at that human enzyme, for example, if we lower the temperature, typically what happens when we lower the temperature of the molecules will simply slow down. And because they're slowing down, the reactive rate is also going to slow down. So how much they react is going to slow down. And then when the temperature is really low, those molecules are just moving so slowly that they're not reacting at all. So typically for enzymes, when you decrease the temperature, the molecules are simply slowing down and not reacting. And so that's why you see a decrease in the reactivity rate. When you go the other direction, though, as you increase the temperature, what's typically happening is the enzyme is denaturing. So it is unraveling. Those R group interactions are breaking down, and it is changing the shape of the enzyme in the protein. And as it denatures, its active site becomes unavailable, and so it's not going to be able to react. And so typically as you go higher and higher, more and more enzymes are denaturing until all of them have denatured, and then there is no reaction rate at all. And so typically for temperature, as you go colder, the molecules simply slow down. As you go higher, the enzymes denature and no longer function. And so that's why you see a curve that looks like that. So the optimal temperature is going to be where it is at the top, the greatest rate of reaction for those enzymes. If we look at pH, again, every enzyme is going to have an optimal pH. The enzymes that are in our stomach are going to work best at a low pH because we've got all that hydrochloric acid in our stomach, a very low pH in our stomach. But if we look in our small intestines, the pH is higher. It's closer to 8 in our small intestines. So those enzymes are going to be working best at that higher pH. We're going to have different enzymes in our stomach than we are going to have in our intestines. And they're going to have an optimal pH. If you go higher or lower from that optimal pH for those enzymes, they will start to denature, and they're going to denature more and more and more as you go farther and farther away from the optimal pH until all of them have denatured and there is no reaction rate. And that's why you're going to see a curve that looks like this for your pH. And so when enzymes and proteins denature, they are going to unfold. Those R group interactions, those quaternary and tertiary structures are going to break down, and the R group interactions will break apart, and the enzyme will unfold. And then it's going to lose its shape. If it loses its shape, it loses its ability to function. If the conditions are changed slightly and you put them back in the optimal conditions, oftentimes those enzymes can renature. But if you go too extreme, temperature or pH, it can be permanently denatured. So sometimes denatured proteins can renature, and if they do renature, they will regain their activity. But if they are permanently denatured, then you cannot put them back. So if you think of it frying an egg, when you go from that clear egg white, which is mostly protein, to that cooked egg white, you can't go back. And that's because those proteins have been permanently denatured. And so if you go too extreme, you will not be able to renature them. Some enzymes have cofactors, and a cofactor is a non-protein enzyme helper. It can be inorganic, such as a metal, or it could be organic, but those are going to be molecules that will help the enzyme work better, work better. And then coenzymes are simply organic cofactors, and those are called coenzymes, and that would include vitamins. Enzymes also have inhibitors, and inhibitors are going to be molecules that will slow down the reaction rate. So a competitive inhibitor is going to be one that binds to the active site of the enzyme, and it competes with the substrate, so the reaction rate slows down because it's interfering with the ability of the substrate to bind to its active site. And then there are also non-competitive inhibitors that are going to bind to another part of the enzyme, which causes the enzyme to change its shape, which makes the active site less effective at binding with the substrate, and that's going to reduce the reactive rate of it. And some examples of inhibitors would include toxins, poisons, pesticides, antibiotics. Those can all be inhibitors of enzymes. So here it just shows you normally for a normal enzyme, that substrate will fit into the active site of the enzyme. In a competitive inhibitor, the competitive inhibitor also fits into the active site and it blocks the substrate from fitting in there. And so the reactive rate is going to slow down. But with a non-competitive inhibitor, it binds to another location on that enzyme, changes the active site so that substrate doesn't fit in there as well. And so that's going to reduce the reactive rate as well. So cells can regulate their enzyme activity, and that's going to save some energy. It's going to be able to turn on or off genes that will encode specific enzymes, or it can regulate the enzyme activity itself. So a cell that's efficient can actually turn on and turn off its enzymes and regulate how much enzymes are being used and produced. So some ways that enzymes are regulated is through allosteric regulation. And allosteric regulation could either inhibit or stimulate an enzyme's activity. And so if we look at allosteric regulation, we've got, again, activation and inhibition. And an activator would be a molecule that would stabilize the active form of an enzyme and activate it and basically turn on that enzyme. And an inhibitor would be one that stabilizes an inactive form of the enzyme, so basically turning it off. So if we look at that first one, an activator here in green binds to an enzyme, locks its active sites open, and then it makes it more efficient at reacting. So basically, you're turning on that enzyme. And then with an inhibitor, it is binding to the enzyme, and it keeps those active sites closed so that enzyme cannot react. So basically, you're turning off that enzyme's activity. And then sometimes the substrate itself is the activator. And so once one substrate binds to an active site, it locks open the other active sites, and then it stabilizes that molecule so it is more active. So the substrate itself is basically turning on that enzyme. So cooperativity is a form of allosteric regulation that will amplify the enzyme's activity, where the substrate is the one that is the activator. When we're looking at drugs kind of work that way as inhibitors and activators of different metabolic pathways. Another way that cells can regulate their enzyme activity is through feedback inhibition. And so a cell again is going to be more efficient if they can regulate when it uses enzymes and when it doesn't. So if you can turn off your metabolic pathway, you can actually save some energy. And that's what feedback inhibition does. So if we look at this diagram here, here's your metabolic pathway. And we're starting with this initial substrate, threonine, and that is converted through a series of chemical reactions to produce isoleucine, which is the end product. But we have a whole bunch of enzymes that are working along the way to convert threonine into isoleucine. So that's kind of like our factory. It's churning out a whole bunch of isoleucine. But when you have enough isoleucine built up, what can happen is that isoleucine can actually bind to one of the enzymes in that metabolic pathway and shut it down. So you're allosterically regulating that enzyme. You are binding the isoleucine and inactivating or shutting off that enzyme number 1 in the metabolic pathway. That's going to shut down that whole metabolic pathway, and it's going to stop making isoleucine. Until we need more isoleucine, then the metabolic pathway will start up again because you're using up all the isoleucine, including that one that will pop out of the binding site, and then the metabolic pathway will turn back on again. So the end product becomes the allosteric regulator, the inhibitor of enzyme number 1 in that metabolic pathway, turns off the factory and saves some energy until more isoleucine is needed by the cell. And that is feedback inhibition, turning off that metabolic pathway to save some energy, where the end product is the allosteric regulator. I hope that was helpful. That's the end of our discussion on metabolism and enzymes.